All right, so we're going to start, obviously, with the structure of nucleic acids, what they consist of, and what types there are. Now, the building block of a nucleic acid, as we consider it, is a nucleotide. Nucleotide contains three parts. I'm actually going to go back here. So nucleotide contains the sugar, obviously, ribose or deoxyribose, nitrogenous base, and the phosphate residue. The bases are divided into two groups: pyrimidine bases and purine bases. The molecule that you see on the left is the molecule of pyrimidine, which contains two nitrogens in its aromatic ring, and the molecule on the right is the molecule of purine, which contains four nitrogens in the aromatic system. Now, the five bases that you're going to find in DNA and RNA are modified purines and pyrimidines. There are in DNA, the purines are adenine and guanine, while pyrimidines are cytosine and thymine. In RNA, the only difference is that instead of thymine, which is also 5-methyluracil, the RNA molecule uses uracil, which is labeled with the letter U, not surprisingly. So these are bases that are found in DNA and RNA. You can see that pyrimidine bases share that same ring in the center, okay? And while cytosine is found in both DNA and RNA, uracil is found in the RNA only, while thymine only in the DNA. And the difference between the uracil and thymine is this. Purine bases, on the other hand, are universal for DNA and RNA. Now, if you are wondering whether or not you need to memorize the structures of the bases, the answer is no, you don't have to do that. You should be able to recognize which base is purine and which base is a pyrimidine. Clear? Okay, so basically one six-member ring pyrimidine is a condensed fused six-member and five-member rings purines. Now, as we already figured out, there are also pentose sugars involved. So you may remember pentose means there are 5 carbons. In the molecule of RNA, the sugar that is found in each nucleotide is called the ribose, while in the DNA it is called deoxyribose. Since deoxyribose does not have an oxygen on the 2' carbon. Now, these are structures of pentose sugars that are found in RNA and DNA. And the place in question is this. This is the hydroxide substituent at the 2' carbon, while this is just a hydrogen. This difference that may seem minimal, right? Just one oxygen, not there, actually defines, to a large extent, the different properties of DNA and RNA in the cell. Now, I don't know if you wonder why the numbering of carbons in the sugars using this prime, this apostrophe. So let me go back here. You need to number members of the ring, right? The molecule, the carbons in the molecule. You need to number it to show where substituents are. For instance, as we mentioned, this is the 5, sorry, just 5, not prime, 5 methyl group, 5 methyl uracil. So it turns out the numbers that do not have apostrophe, the numbers without the term prime, are used for nitrogenous base. So the numbers that are used for carbons in pentose sugars, they do have that prime annotation. All good? Any questions? All right. So the combination of a sugar and a base is a nucleoside. Okay. Now, nucleoside is produced when one of the nitrogens in the pyrimidine or in the purine forms a glycosidic bond, so-called N-glycosidic bond, to the first carbon of a sugar. And the naming goes that for purines, the ending changes from INE to OSINE if it's a purine. If it's a pyrimidine, first two syllables usually, and IDINE. Let me exemplify this. So here you have adenine, you have ribose. This nitrogen loses hydrogen that was attached to it. OH group leaves first carbon, and the beta N glycosidic bond, beta because OH group in this case looks up. Beta N glycosidic bond is formed. The name goes from adenine for a base to adenosine for an entire nucleoside. I'm going to highlight INE. Questions about the naming? I'm going to provide you with sort of a cheat sheet here. So we go, adenine, adenosine, guanine, guanosine. These are purines. Now for pyrimidines, it's a little bit more wonky compared to pretty strict naming rules for conventional organic compounds. So thymine, thymidine, uracil, uridine, cytosine, cytidine. You see a little bit different, okay? But overall, you can kind of follow the logic. These are pyrimidine bases. Now, when a nucleoside gets combined, it forms what effectively is an ester with a phosphate group. This compound we call nucleotide. The phosphate group always bonds for a compound being called a nucleotide. The phosphate group has to be bonded on the carbon 5. Whether it is a ribose or deoxyribose, the bond that is formed is effectively phosphate ester bond. Now, can other hydroxyl groups on ribose or deoxyribose form that phosphate ester group? Yes, they can. But in the cell, nucleotides exist as 5' monophosphates. Only 5'. So these are 5 nucleotides, actual building blocks of nucleic acids. The full name will include the name of the nucleoside, adenosine, okay, and location, 5', and monophosphate, meaning that there is only one phosphate group. Based on the fact that we use monophosphate, you can easily imagine that there can be di and triphosphates. We're going to talk about that as well. We doing all right so far? So you can move on and talk about guanosine, 5 monophosphate, uridine, 5 monophosphate, and so on and so forth. I can highlight a slightly different thing here. For instance, cytidine monophosphate, cytosine base, can be found in both DNA and RNA. If the sugar, the ribose, loses OH group and becomes deoxyribose, the full complete name is deoxycytidine 5' monophosphate. Do you understand where deoxy in front of the nucleoside name is coming from? Does that make sense? Let me re-exemplify it again, okay? So, if the sugar is ribose, then we call it adenosine monophosphate. If the sugar is deoxyribose, then we call it deoxyadenosine monophosphate. Which brings me to the summary of what we just discussed. Okay, so this is the summary of components. You should know the difference between the DNA and the RNA in terms of the composition. In RNA, there's U instead of T. And there's a ribose instead of deoxyribose. So the full name, as we discussed, considers whether there's a hydroxyl group in the 2' position, and you need to specify that there's monophosphate on the 5' carbon of the sugar. To be honest, nobody really writes this except on the cans of, like with the reagents. When people need to say, we're going to synthesize DNA and we're going to have certain nucleotides, we just use single letter, okay? No, but like you can say, now we're talking about nucleotides and just use letters. If you need to talk about nucleosides, you can also say, now we're going to talk about nucleosides, we're going to use the same letters, but they're going to mean slightly different for that particular conversation. Okay? I'm not going to read the table for you. This is the summary again. So let's move on. Okay? And the bases, components, so guanine, DNA or RNA, or both? Both. Adenine. Exactly. Thymine. This DNA, yes, that is correct. And uracil, that's RNA. So, how are nucleic acids organized? DNA and RNA have fairly different structures, and we're going to learn about those structures. But the basic idea is that nucleic acids are comprised of many nucleotides forming a chain. Okay? In this chain, 3' hydroxyl of the sugar in one nucleotide bonds the phosphate on the 5' carbon in the sugar of another nucleotide. So that creates a unique sequence of bases. And that sequence, just like with the proteins, the sequence of bases is what's called a primary structure of a DNA or RNA. So obviously any chain has two ends. So one end of that chain will have a sugar with a 5' phosphate that did not react with any other molecule. On the other chain, there's a 3' hydroxyl group. So it's better to see in the image. Take a look. So this is OH group on 3' carbon. It reacts with one of the oxygens on the phosphate group. Okay, forming phosphodiester linkage. So now, when you start looking at the molecule, you can clearly see that the 3' carbon is connected to the phosphate of another nucleotide through phosphodiester linkage, and that phosphate was hanging off of a 5' carbon of another nucleotide. Now, that means you have a sequence of nucleotides in the DNA, and they all are oriented in the same way. Does that make sense to you? Yes? Okay, so first of all, it's always, the phosphate always connects 3' carbon of one sugar to 5' carbon of another sugar. The direction is always the same, 3 to 5, 3 to 5. Okay? And if you look at the phosphates and the carbons, they go phosphate, not carbons, sugars, phosphate, sugar, phosphate, sugar, phosphate, sugar. Can you see that? This order forms what's called sugar phosphate backbone. It does not carry any information. Kind of jumping ahead, but you understand that it doesn't carry any information. What it does carry, what carries the information is the sequence of bases. Now, obviously, people do not, do this massive drawing of formulas, because it doesn't really matter. What matters is the order of bases in the molecule of DNA. This is what's going to be the signal. So when we write, now hear me out, we're not going to go through that learning check. When we write the sequence, it's a convention. We write it so that 5' end is on the left, 3' end is on the right. That's it. We agreed on that. Okay? Does that make sense to you? Yeah? Yes? It's a convention. There's no deep biological meaning in that particular writing. So in this case, the base on the 5' end is cytosine. Now, first we're going to talk about DNA, and we're going to discuss its secondary structure, in other words, a double helix. So, in the 1940s, yes, the biologist named Michael Chargaff formulated the Chargaff rule. He discovered, experimentally, he was a biochemist, that the amount, if you isolate DNA molecule from a nucleus, and by the way, this is why we call it a nucleic acid. If you isolate that DNA molecule from a nucleus and try to determine the amounts of each individual nucleotides were known by then, you will discover that the amount of adenine is the same that amount of thymine, and the amount of guanine is the same as amount of cytosine. And that, effectively, what happened, number of purine bases was the same as the number of pyrimidine bases. Now, this stack of slides does not actually explain the logic of that discovery. So that was first, that is here, it's called the Chargaff rule. But what about it? So now let me give you the whole story. So there were two camps, one that was led by Watson and Crick, and another camp, quote-unquote camp, that was led by Nobel Prize winner Linus Pauling. Okay? They had drastically different views on the hypothesis of the DNA structure. And by the time it was pretty well established, well, not well established, but it was discovered that DNA does carry the genetic information. So the scientists at the moment were equipped with Chargaff rule, the one that I just described to you, okay? And the fact that DNA stores information. So the question was, how does it work? A woman named Rosalind Franklin, who was working with a guy, this is purely science history story. So this is just for you to be aware. Rosalind Franklin was working with a guy named Morris Wilkins, and they hated each other. They did. Mostly because of the fact that Franklin was a proponent of Linus Pauling's model. And Wilkins, together with Watson and Crick, were proponents of a different model. Franklin did an X-ray crystallography. It's basically when you make a crystal and shine the X-ray through the crystal, and then you have some kind of a picture, and then you try to deduce the shape of the molecule based on that picture, because differently shaped molecules will produce different pattern on the film. So she produced a very famous image of that X-ray, and her results were stolen, used without her permission by Wilkins and Crick and Watson. So Wilkins took those results without asking her, without crediting her. That's important. Okay, she never got credited, and gave it to Crick and Watson, and based on that result, which was very characteristic for double-stranded helical molecule, they built this model. There is a seminal 1953 paper in Nature, which is 1.5 pages. That's it. That proposed the DNA structure, okay, as a double helix. So they suggested that there are 2 strands of nucleotides. They wind around each other as a spiral. The sugar and the phosphate form the backbone. Bases are arranged as steps inside. And what's important, the strands were anti-parallel. One strand goes from 5' to 3', and the second strand goes into the opposite direction. So they also demonstrated, and it was quite amazing, there's a famous photo of them too, they also demonstrated that structurally this pairing is the only one that is, not the only one, but this pairing is the best. Adenine pairs with thymine, 2 bases, and guanine is paired with cytosine. Okay? So that forms those famous pairs AT, GC. Now, what holds those strands together? What linkages are established between this bases? Linkages are hydrogen bonds. You can see them here. And as you may remember, while hydrogen bonds are weak, there's a number. It's all about the multitude of them. So they're strong enough to hold 2 strands of the helix. The pair between the G and C is stronger because there are 3 hydrogen bonds, unlike 2 between A and T. So this is the classic representation of a DNA molecule. You can see that there are 2 strands. I'm going to call them, for the purpose of our conversation, left and right, which is not correct, but okay. So you can see that left strand has a 5' end on top and 3' end on the bottom. The ribbon here represents sugar phosphate backbone, and the right strand has 5' end at the bottom and 3' end on top. So strands are anti-parallel. Does it make sense so far? Okay, good. So complementary strand, what pairs with A in the DNA? G, T, C, G, T, T, A, G. Now, if we add the ends, again, hear me out. This is the convention. There is no massive biological sense in how you orient the sequences on paper. But by convention, this is 5 prime. On the top strand, this is 5, this is 3, and on the bottom, they are anti-parallel. Am I clear? All good? This is how nerds decided to write this stuff. Why? The idea that DNA molecules have directionality. Why is this idea important? So it's important because it gives DNA the ability to store the information. So let me ask you this. When you read in English, in which direction do you read? Left to right. If you read in Hebrew, or Arabic, it's going to be right to left. Okay, make sense? Yeah? But the point is, neither of the languages, not English, not Hebrew, not Arabic, they do not have words in which, I'm going to read this word left to right and then the next one right to left. So the fact that DNA is oriented, like 3' to 5', means that you can consistently read the message. You can write the message by DNA. And that double-stranded structure also enables DNA to be able to replicate, to be able to reproduce, to be able to get reproduced by the enzymes of the cell. So during the DNA replication, which goal is to pass the genetic information from the parental cell to the daughter cells, in this process, the strands of the parent DNA, they separate, and each individual strand is independently copied using the principle of complementarity. Okay? So the classic illustration of that process is, let me see if I can squeeze it in here. So imagine you have a DNA that has two, we're going to call them blue strands. Okay? So these blue strands are separated and new red strand is added. Okay, makes sense? This is called semi-conservative replication. If you will take a biochemistry class or molecular biology class ever, you will be talking about famous Michelson-Stahl experiment, which was done by Michelson and Stahl, and they literally discovered, like they said, there are several mechanisms, these are the outcomes of our experiment that would match each mechanism, perfect hypothesis, and the resolution of the issue. Okay? So how can it happen? So first of all, you need to break those hydrogen bonds. This is the function of the enzyme called helicase. It separates the strands of a double helix by breaking the hydrogen bonds. Each strand is a template for the synthesis of a new strand. So here you can see the separated strands, okay? You can see the addition of new nucleotides, okay? Does that make sense? You see the addition of new nucleotides, and the blue strands, unlike not the purple strands, are the new one. So that's a new strand, and this is the old strand. Okay. So the enzyme that is essential for that process, for the process of synthesis, well, they all are essential, but the enzyme that actually does the addition of the new nucleotides is called DNA polymerase. More specifically, DNA polymerase 3. There are several DNA polymerases. So what effectively is the product of that process is two new, I will reformulate that, because what we effectively get from one double-stranded DNA molecule, you get two double-stranded DNA molecules. Make sense? And that's your daughter DNA. So the reason why this is possible is because the bases are complementary. When DNA polymerase, look, I'm gonna write down the old strand first. Let's say it's CGTA. Okay, the synthesis always goes, the reading goes from 3' to 5'. So when DNA polymerase starts reading this, it goes, okay, what should I put against C? It's going to be G. C, A, and T. And that's your new strand. Okay, make sense? Now, I mentioned several times, we're going to get into transcription, that DNA is a carrier of genetic information, probably the most important molecule, right, in the cell. What is the biological importance of having two strands? If you have one strand, can you restore the second?
No.
Dude, I just did. Look at the red one. Let's say I have a red one, right? I have just one strand that is the old one. Can I make it double-stranded? Yeah. I know exactly which bases to put against which. Two strands, one is a backup. Well, technically speaking, they both are backup. If one gets damaged, another strand can provide the unadulterated sequence, unchanged sequence, unmodified sequence, and restore the integrity of the damaged strand. Does it make sense to you? Now we're getting into more of a protein synthesis process. We're going to talk about RNA, and we're going to talk about transcription and potentially translation. So, should I do it now? I probably should. Okay, so there is an analogy which I came up with, okay, and I'm going to give it to you. And I'm going to explain it to you, and the understanding of the whole protein synthesis process will become a breeze, I promise. So what is DNA? DNA is a recipe book. Okay, it contains thousands and thousands. Human DNA, it's probably 20 to 25,000 recipes. Okay? Each recipe equals a gene. Now, imagine your friends call you and tell you that they're about to come over for dinner. And you start thinking, okay, what am I supposed to cook, right? So think, okay, I have one friend who's lactose intolerant. I have one friend who has celiac disease, one friend who's vegan, another friend is, say, Jewish. So you think, okay, nope, one food without pork, because Jewish, right? Something without gluten, something without milk, and some vegan options. So you concoct some kind of, menu, right? And then you look at your book, 25,000 recipes. If you have an actual recipe book with 25,000 recipes, will it be massive? It will be. Also, remember, it's your family heritage. It's the most important book in your house. Are you going to take it into the kitchen? Well, all this shit can happen. Probably not. Also, handling 15 recipes out of 25,000 that may be all over the place, maybe, probably not a good idea. Want to make a copy of a recipe. Does that make sense? So, how do you make a copy of a recipe?
Do you need one?
You literally have a book. I'm asking literally about the book. You have a book of recipe. You don't want to take it into the kitchen. How do you take a recipe into the kitchen, though?
You just copy it.
Write it down, or better yet, snap a picture with your phone, right? So that picture or the copy of the recipe is RNA. Does it make sense? That this is not food. You can't feed people with like, hey, look, I have a picture on the phone, right? You need to make a dish. You use the instructions to make protein. Okay? Sorry, I don't need an arrow here. But now, think about the implications of all this story. The book, it's very stable, right? DNA is stable, okay? The copy, you don't really need this photograph after you finished, right? So RNA is unstable and it degrades fast. And your dish, this is what you're feeding to people. Proteins are main functional molecules. By the statement functional, I'm not implying that DNA and RNA have no function at all. I'm saying that 99% of cellular functions at the end of the day are proteins, signaling, enzymatic activity, structural activity, all of that. Okay? Does it make sense to you? And another thing, that book that you have, there are no mistakes. It's very error-proof. When you write down a recipe, imagine that you copy a recipe on a piece of paper and you bring it to the kitchen and it turns out you have or you made a mistake. You wrote 5 tablespoons of sugar instead of 1 tablespoon of sugar. It comes out as an absolute crap. Is it a problem? No. You go back, you make a copy, a new copy, which means making RNA is, you don't need that reliability. It's error-prone. Okay? Now, let's go back to this conversation. So what is the difference between RNA and DNA? Well, first of all, ribose instead of deoxyribose. bases. Instead of thymine, you have uracil. RNA molecules in the cell, for our intents and purposes, are single-stranded, unlike DNA, which is double-stranded. And RNA molecules are indeed much smaller than DNA molecules in the cell. Okay, overall, DNA is bigger. Three main types of RNA molecules that can be found in the cell is the messenger RNA, first of all. So that illustration here, I missed one detail. There should be letter M in front of RNA, because that recipe copy is the messenger RNA. Messenger RNA just transfers information, okay? So it carries information from the nucleus to the ribosomes in the cytoplasm. When I talk to students about this, I tell, DNA is in the nucleus. Nucleus is your living room. Not a lot of action happens there. Ribosomes are your kitchen. This is where proteins are being synthesized. Transfer RNA or tRNA, its role is interpretation of genetic information and the transport of amino acids to the site of translation, to the ribosomes. And finally, ribosomal RNA, or rRNA, it is the part of ribosomes. So this table summarizes different types of RNA in us, and it also provides you with the percentages. Please do not memorize the percentages. Let's jump to the structural features. So first of all, let's talk about tRNA. All tRNA structures are described usually as the cloverleaf structure. Okay, now, to be absolutely fair, it kind of does look like a leaf of clover, right? You see what I'm saying? Okay, so in reality, on the right, the image B shows you the actual structure of tRNA. It's not a clover leaf. But what's important, there are a couple of really critical parts. One is the acceptor stem, and another one is anticodon. We're going to talk about codons and anticodons when we get to translation. So the acceptor stem here is where amino acid is getting attached. Okay? The anticodon loop is the part of the RNA, tRNA, that recognizes, a codon. I will illustrate it in a few minutes. So this will be codon recognition. Now, we mentioned several minutes ago that RNA molecules are single-stranded molecules, and yet you are looking at these structures, and they definitely appear like having this double-stranded parts, right? So what's going on here? These are intramolecular hydrogen bonds. So they form intramolecular double strands. But if you break all the hydrogen bonds, if you denature tRNA, it's still one strand of nucleotides. Am I making sense? Just FYI, in terms of structure naming, this intramolecular double-stranded part is often called a hairpin. And guess how this is called? It's a loop. So you're basically seeing here 3 hairpin loops and 1 just hairpin. So what is the function of this tRNAs? How they work? Let's first describe. And I'm using my phone to Google. I don't remember the genetic code table. Let me see. I'm gonna pop. Okay, that's good. So let's say, first of all, you have 20 amino acids to code. Okay. Imagine that you have 20 objects in the world. That's it. 20 amino acids. Do you follow my train of thought? You need to develop language that would allow you to name all 20 amino acids. Do you follow? So your language has 4 letters. If it's DNA, A, T, G, and C. If it's RNA, it's AU, G, and C. Does that make sense to you all? Yes? Okay, so I'm going to be using RNA language because I have that genetic code here, but it's not really different. Okay? So 20 amino acids. Can you name each amino acid with a single letter? No, because then you name 4 amino acids. What about others? You can't give the same name. Make sense? If you will try to use 2-letter words, you will have only 16 combinations. Okay? Does that make sense? So you have to use 3-letter combinations. They're called codons. Okay? Am I making sense? So I'm going to write a completely bogus hypothetical sequence, RNA sequence, okay? So we have A, U, G, and I'm going to break them. Do you understand what I'm saying? Like I'm going to separate them. So this is first word. This is second word. This is third word, and so on and so forth. Okay? So how does a tRNA recognize which amino acid it should incorporate into a protein? Well, ACU is the code for an amino acid threonine. Does that make sense? So the tRNA molecule, I'm going to use the schematic representation as a cross, okay? That tRNA molecule is going to have its anticodon complementary to ACU, U, G, A. And on the acceptor stem, there's going to be an amino acid threonine. Does that make sense? So this is the codon, and this is anticodon. And by analogy, if you use that exact logic, which tRNA will recognize this. Okay, if this is the codon, then anticodon should be U, A, C. Would you agree with me? Yes? Because it's complementary. Okay. So what will tRNA carry on the acceptor stem? AUG is the codon for an amino acid, methionine. Okay? Does the concept of codon anticodon make sense to you? No?
So my question is it basically, we're pairing it, right?
Say it one more time.
So we're basically pairing it to get the anticodon?
Anticodon must properly pair with the matching codon.
Okay. And so we basically put in, okay, I get it, the A.
Like if you will try to stick threonine over here, like if that tRNA will try to bind to AUG, it can't, because it's not fully complementary. You have to have a full complementarity. Does it make sense? Awesome. So I have serious issues with these statements, but so protein synthesis of protein is translation. Transcription is the synthesis of RNA, okay, to carry the genetic information to the ribosomes. Am I clear? So for your sort of future reference, because folks, some stuff here, like, I have to use it since, I'm not the boss, kind of, but the better name, gene expression. This is your cooking, okay? It does involve transcription and translation. And translation is the protein synthesis. So this shows you 2 steps here, okay? You have transcription that happens in the nucleus. And then translation that happens in the cytoplasm by means of ribosomes. Basically, the whole idea is to carry the information by mRNA and then interpret it using tRNAs to make a protein. So when your DNA is being transcribed, remember, you are taking pictures of the recipes or copying it. So DNA unwinds, and the enzyme called RNA polymerase uses a strand of DNA as a template. And the principle of the synthesis is the same, complementary base pairing, okay? So if there is an A in the DNA strand, again, across from it in the RNA there will be a U because it's a U pair. If there is a G in the DNA strand, it's going to be C. If it's a T, it's going to be A because AT pair. Okay? And if it's a C, it's a G. Okay. So now let's take a more holistic view of the process of transcription. So here you can see the enzyme called RNA polymerase. This is the enzyme that does the entire process. There's a different number of So in human cells, there are 3 different RNA polymerases. I remember that RNA polymerase 2 is the one that actually synthesizes mRNA. RNA pool 1, pool 3, they are for like ribosomal RNA and tRNA. I don't remember which one is which. What's important is this. So first of all, take a look. You need to split the DNA strands. Okay? Does it make sense? When they split, this split thing is called a transcription bubble. Then there is a copying. You can see mRNA being produced here. How many strands of DNA are being copied? How many strands serve as an actual template? It basically shows you this. Like there is a statement, there's a writing on the screen that tells you how many template strands are during transcription. On the left, what does it say? There's one strand that is a template, and then there is another strand that's called informational or non-template strand that is not being used. Does it make sense? So this bottom strand here, 3' side on the left, okay? It's 5'. I traced. That's the correct one, okay? And then there's a informational non-template that is not used. So this is a very interesting story about transcription. Only one strand is transcribed. One RNA strand is synthesized. So, here's the question. Can we just take one strand of DNA, just one, and make a copy of it in the cell using RNA polymerase?
Do you know?
No. The answer is no. It's one of the fundamental laws in nature. It actually is a fundamental law that double-stranded DNA is required. So you have to have 2 strands. One is not just going to be copied, that's it. We're doing all right? Okay. Now, so if we have a template strand with that sequence, we're not going to pick A, B, or C. We're just going to write down the strand. Am I making sense? So what should be across from C? No.
No, sorry. C or A?
Nope. That's G, T,
A.
A. Remember, it's RNA.
So it's U.
It's U, G. So that's your answer. Okay? Now, next sort of logical stop now is to talk about genetic code and translation. So first, genetic code. So genetic code, as you already know, I already mentioned that, consists of codons. Okay? So these are mRNA codons. In this case, the codon UUU encodes the amino acid phenylalanine. Okay, you can see that the codons do not overlap. Now, since every codon is 3 amino acids, since every codon is 3 nucleotides, okay, 3 nucleotides, it means that there are 64 possible combinations. Okay, how many amino acids do we have? 20. So we have more words for our 20 objects. We have a special word, start codon, to signal where the protein synthesis should start. And we have 3 words as stop codons to signal where the synthesis should, well, stop. Am I making sense? But even if we take those 4 codons out, we still have 60 more. So I want you to take a look at this, the table of genetic code. Please memorize it completely. I'm just joking. It will be actually provided for exam 4 in the final. Okay? You will have it if you need to decode something like that. So first, let's identify the start codon. You see that the start codon also serves as the codon for amino acid methionine. So one problem is solved. Then we have stop codons. 1, 2, 3. Okay? Make sense? And we have 60 codons for now 19 amino acids. That's a lot. So it turns out that the same amino acid, let's take an amino acid serine, it can be encoded by more than one codon. Okay? So, if we look at this connection, one amino acid can be encoded by few codons. Not necessarily there is tryptophan, which is only one codon, but every codon encodes only one amino acid. This feature of genetic code, that it has synonyms, okay, is called, the genetic code is redundant, or another term, degenerate. Does it make sense? Language-wise, it's very easy to explain. Let's say you have never seen an actual elephant, and you ask me about the size. Can I describe an elephant as large? Can I describe it as huge? What about enormous? Yeah, those are synonyms. But if I tell you that the elephant is huge, you understand it's not going to fit in your pocket. Like, you understand the huge has only one meaning. The same way, amino acid can be encoded by several codons, but if you take a single codon, it always means the same amino acid. UUU will always encode phenylalanine. Okay? CUC will always encode leucine. So this, just shows you, basically, can you find combinations of 3 letters in the table? I believe you can. Okay? So it just shows you that if CCU is codon for amino acid proline, proline will be inserted into the polypeptide chain, and so on and so forth. Will you be able to do that? So this is the exercise that I think is best suited for 4-year-old, which means, find a combination of 3 letters that matches another 3-letter combination. GCC, okay, is here. That's alanine. So the first amino acid here will be alanine. GUA is valine. And GAC is aspartic acid. So this. Will you see it on the exam? It is possible. Will you be provided a table? Absolutely. So you should be able to do something like that. So basically, during translation here, you can see that tRNA that has this anti-codon binds to the corresponding codon in mRNA, and the peptide bond is formed between the incoming amino acid and the polypeptide that sits on another tRNA. Now, once the amino acid is bound to the synthesizer chain that's being synthesized, this free tRNA gets out of the ribosome and picks up another one. Okay? Does it make sense? Basic concept. Believe me, we are omitting a lot of detail. But general idea, does it make sense? Okay? So that these tRNAs, in a very specific manner, bring amino acids in. So they are added exactly according to the code, exactly according to the sequence of 3-letter words that we call codons, and that sequence is the sequence of messenger RNA. Okay? Does that make sense? So when the ribosome, that big thingy here, when the ribosome reaches the end of the gene, the end is marked by a stop codon. Instead of tRNA, there is a so-called release factor that separates polypeptide and the ribosome and then modify it somehow. Okay? Does that make sense? So now you have a chain of amino acids. Because amino acids will start forming hydrogen bonds between carbonyl and amido groups, okay? They're going to start folding into secondary structures, okay? And then within those secondary structures, there will be salt bridges, disulfide bonds, and eventually that's going to be tertiary or quaternary structure. So at the end of the day, this is your functional protein. Now, do I have sort of an analogy for this primary, secondary, and tertiary structure? I do. Hear me out. Let's say one of the dishes that you're about to make is mashed potatoes. Okay? So what do you need for mashed potatoes? You need potatoes, you need milk or water, usually, right, to make it fluffier, usually milk, and you probably need some salt, and some spices, whatever. Okay? Do follow my train of thought. So first you get the potatoes out, okay, you put the salt on the table and you put some milk on the table in a cup. That's your primary structure. You collected all the components. Then you peel the potatoes and boil them, okay? And you heat up the milk and you add some salt. That's your secondary structure. It's about to be ready. Tertiary structure, you mix milk into boiled potatoes, right? Mash them. That's your tertiary structure. That's your ready-to-go meal. But you're probably going to combine mashed potatoes with something else, like, I don't know, meatloaf and gravy. Those are two other proteins, and the meatloaf protein and the gravy protein and the mashed potato protein, they form a final meal. That's a quaternary structure. Okay? Does that make sense? So, okay, this just shows you that during transcription, that strand is copied into mRNA, and this shows you the codons on tRNAs, and that's the final sequence of a protein. So what is the importance of us knowing that stuff is very hard to overestimate. For example, a lot of antibiotics, they actually block not only protein synthesis, but even those processes, even replication. So some examples: chloramphenicol inhibits peptide bond formation. Erythromycin prevents the ribosome translocation, so it can't move from one codon to another. Puromycin, incomplete protein is released, okay, early termination. Streptomycin, proper tRNA attachment is blocked, and there are some faulty proteins that formed, and tetracycline prevents the tRNA binding to the ribosome. Even more than that, hear me out, even more than that, there's an antibiotic that inhibits transcription, called the group of antibiotics called rifamycins. Now, the last bit to chat is a recombinant DNA. So what does that mean? It means that the DNA from one organism is combined with DNA from another. So for the most part, the idea is that you bring the gene of interest and insert it into a so-called DNA plasmid in bacteria called E. coli. Okay? So let's jump straight to the image. So imagine that you have these plasmids. Plasmid, hear me out, plasmid is a circular double-stranded DNA that is not essential for bacterial cells. It can be forcibly inserted into a cell. And if the plasmid carries genes, these genes are going to be expressed. The corresponding proteins are going to be synthesized in that cell. Am I making sense? No? So imagine you have a cell, E. coli, right? So this is your bacterial cell, right? And this is the DNA of E. coli. There's no nucleus, so no nucleus. What you can do, you can take a small circular DNA, the plasmid. Let me see if I can make the circle perfect. Yes. And force it in here. Okay, so now it is inside the cell. So genes from the plasmid going to be expressed. And the corresponding proteins are going to fill up bacterial cell. The idea of a plasmid, does it make sense now? So imagine you have that plasmid, and you use so-called restriction enzymes to cut out a bit. Now, if you look very closely, you can see that the cutting produces what's called sticky ends. You see how they pop out, stick out, right? So these sticky ends are also generated on the DNA fragment, the gene of interest. You mix your plasmid here, and you mix the gene of interest, and they stick to each other because the ends are matching. Okay? You treat them with the enzyme called ligase, force that plasmid into DNA, into bacterial cells, and these guys now, first of all, you can grow a buttload of those bacterial cells, and they will be producing new protein. Okay? Does that make sense? Now here's the question. Can you find some gene that produces some horrible toxin and clone it into a molecule, into plasmid, and put it in E. coli, and it's not going to be very bad? You can, right? And you can accumulate large amounts of bacteria, and if there's some kind of like you spread it somewhere, you may harm people. When this technology was established, the scientific community by themselves put a stop on all experiments with recombinant DNA because they realized the potential gravity of consequences. They held a conference, so-called famous Asilomar conference, in which a bunch of nerds got together and figured out the rules. And since then, experiments with recombinant DNA, for the most part, well, they all are guided by those rules, and they are reviewed by the committees of nerds at each institution to make sure that it's not something like horrible. Does that make sense? I can tell you that as a person who worked in science, science actually is pretty good in self-regulating. So why this is cool? We can clone a gene into a plasmid, put it in the bacteria, and produce enormous amounts of various proteins that we want, such as insulin, erythropoietin, human growth hormone. I'm not going to list this. I'm not going to read this table for you. Does that make sense? I'm going to correct some stuff here. So, first of all, influenza vaccine is not produced. It's not recombinant. I can guarantee you as a person with a lot of virology experience, absolutely not. No. It's just mistake. It's a mistake. And for the most part, hepatitis B, I don't know if it's a bacterial, but we and bacteria, humans and bacteria, are very different. Like biologically, very far from each other, evolutionary. So now it's not as much bacterial cells. It's mostly done in yeast. But the concept is very, very similar. Okay? Okay. So another important implication of knowing all that stuff is that we humans established polymerase chain reaction, PCR. Remember how you can copy individual strands in the process of replication? This is effectively replication. Okay. So in a PCR, you can in a very specific fashion copy a very particular region of DNA, And when you copy it once, look what happens. You go from one DNA, right? One copy to two. When you make copies of this 2, you go to 4. When you make copies of 4, you go to 8. So the number of DNA copies of a particular region increases exponentially. Does it make sense? Yes? Yeah? So with every copy, you increase it. If you do 20 cycles, you can increase the number of that particular fragment that you're copying more than 1 million times. So this method is extremely sensitive. Also, you can identify an absolutely unique fragment of DNA within the sample. Let's say you're looking for the presence of SARS-CoV-2, the virus. Okay? It's highly sensitive. It's highly specific. And this is why it's so useful for the diagnostic purposes. So that method effectively allows the genetic testing. You can screen people for defective genes, or you can screen people like if you come in and you are sick, respiratory. Now there is so-called respiratory PCR panel. They will take a sample and they will run it in the lab, and it's very quick, can be automated. So sometimes if it's properly organized, by the end of the day you can know what you have in terms of respiratory infection. Okay, it is true that it's used widely in the diagnostic of genetic disorders, such as breast cancer. Okay, basically, anyone did Ancestry or 23andMe ever? No, no. Ancestry and 23andMe, you know, when they look at if people are related to each other, they do PCR, pretty much. Very cheap PCR, okay? DNA fingerprinting. So there are so-called restriction enzymes that can cut DNA. And each restriction enzyme, recognizes a very specific sequence, a place where this DNA is going to be cut. And if you use a panel of restriction enzymes, they will cut the DNA into smaller, unique fragments. That's important. Okay? And then these parts of the DNA, these fragments, can be separated by electrophoresis when the electrical field is applied to DNA fragments, and they move according to their mass. The smallest are moving faster, and the largest are moving slower. Okay? And that bending pattern is unique for people, because we have slight differences in our DNA. Okay? Now, in 2003, Human Genome Project was completed. Human DNA was finally sequenced. It was a very interesting experience because it started in like early 1990s, and it cost about $300 million. So it took scientists more than 10 years to sequence human DNA, and a lot of money. Okay? That make sense? Now, I believe that you can pay for DNA sequencing, and it's $1,500. And it's going to be ready, like your DNA sequence will be ready in a couple of days. So, check the progress. So, we are probably a few years from the time when a newborn will have their DNA sequenced and interpreted, and it will be known what diseases they are particularly sensitive to. Am I making sense? Okay. So only 3% of human DNA actually encode things. So imagine your recipe book, those 25,000 recipes. You have 30 times more if not 32 times more, information that is not recipes, just some gibberish. And that gibberish is regulation and recognition by proteins. So recombinant DNA technology requires the use of, obviously, restriction enzymes and plasmids, but I told you, technically speaking, it's also yeast. Okay? So let's take a break.